Our next speaker is Dr. Ann Summers, PhD. Dr. Summers is a microbial chemist at the University of Georgia. She applies genetics, inorganic biochemistry, biophysics, and computational biology to study the effects of mercury exposure on our molecules, proteins, mitochondria, and metabolic pathways. She discusses the natural defenses the cells can mount to recover from mercury exposure. Please join me in welcoming Dr. Ann Summers.
Thank you. This is going to be, buckle up, this is going to take you back to perhaps your biochemistry or microbiology courses that you took in undergrad. Dr. Cantor's talk, wonderful talk, something you can apply in your practice immediately. This is going to be something to think about in terms of applying in your practice as we go back down, drilling down into real reductionist biology. I hope it's not as painful as your early biochemistry and microbiology courses. And I have a reflex to sort of look out and ask for questions, but I know I can't do that, so I'm going to try not to. But Dr. Cantor's talk certainly set up very much what we're going to be talking about here now, because one of the key aspects of IAOMT's positions has to do with the exposure to mercury. And certainly everything that she was talking about and others talk about here is affected by that premier toxin, mercury. And so that's the focus that I'll have, as well as the microbes, because the microbes used to be ignored in so many areas of biology, human biology. But now we know, as she pointed out, they're everywhere. And we have to learn to get along with them or outfox them when we can. So first I'm going to talk to you about mercury chemistry very quickly and ecology. And then I'm going to talk to you about the system that we've been working with as a bacterial cell as a model for mercury intoxication, and specifically focus on protein vulnerabilities, and then also looking at proteins are the victims, the main victims, not the only ones, but I'll talk mostly about protein as victims of mercury intoxication, and then moving on to something that has to do with the sequelae of mercury exposure in a cell. What does the cell try to do after it's been exposed to mercury? Our cells try to do something after they've been exposed to mercury, and that's a much more complicated story. Bacteria are simpler to work with, but owing to evolution, we can learn a lot from them that will allow us to envision what it is we need to ask most specifically about what's going on with human cells that have been poisoned by mercury, specifically. Eventually, and in fact not too long, we hope to be able to think about how we can support the body's own recovery systems when they've been exposed to mercury, since it'll still be a while before we can get rid of all mercury in dentistry. And there's lots of other mercury exposure. So we'll talk about trauma and recovery, and then we'll switch gears in a big way. Though nature has not switched those gears, nature has put them together. There's this very strong and important connection between exposure to mercury and the development and spread of multiple antibiotic resistance. So that's what we'll wrap up with. So first of all, mercury chemistry and ecology. Remember this? Yeah, this is the modern platform presentation. The group that mercury falls into is group 12, and there are very important elements above group 12. Mercury lies in group 12, which has another very toxic element right above. The point I want to make with this very quickly is that mercury has preferred ligands in the cell. They are sulfur, which is a very important element in all of biology. And so the target at the basic biochemical level of the interaction with mercury is sulfur, like compounds like cysteine and glutathione, and the sulfurs in proteins. So this is an old graph, but I haven't seen a better one since, that shows the limited history of, it's a limited vision of the history, the huge history of the use of mercury in commerce in modern countries, and this starts off, and it was made by the National Institutes of Science and National Institutes of Science and Testing, I think. Perhaps wait, did it again. It's somehow set up as if I touch the touchpad in order to move the in order to move the mouse, it decides I want to change the slide. So this is going to be fun. The use of mercury was hugely batteries, and you can see that in the early 1990s, that there was a big drop-off in the use of mercury in batteries. And so that really helped cut down on environmental exposure. Now, that meant this is to do with the implementation of mercury, the use of mercury. All that mercury is still out there in landfills, okay? It didn't like disappear, right? Prior to that, the use in dental, which is the pale lavender color there, was a relatively minor part, though it was substantial in terms of tons of mercury used annually in dentistry. It now represents pretty much the same number of tons used in dentistry. It's gone down a bit, but it's a larger proportion of people still using mercury lies in dentistry. So mercury gets around. It is around everywhere, and it gets around everywhere. So I'll call your attention down here in this corner right there to the compound mercury sulfide. That's the ore cinnabar, which is the natural ore of mercury. It's a very, very strong association between mercury and sulfur. It's essentially insoluble. Nonetheless, there are bacterial processes which can move that material into methyl mercury, or actually to some degree reduce it to mercury vapor. And so over on that side of the slide, we can see the processes that are involving bacteria in natural ecosystems. And methylation of mercury right now, with a cessation for the most part of the production of methyl mercury as a fungicide, which was the origin of the terrible Iraqi disaster and the Huckabee disaster and so forth, where people mistakenly used beef products or pork products that had been grown on methyl mercury containing grain. That's not done anymore, but the natural source of methyl mercury right now is microbial activity in the environment. And if you're eating fish that has methyl mercury in it, and you are eating fish that has methyl mercury in it because fish does, that comes from natural microbial processes, from natural occurring mercury, which is distributed worldwide to some degree by human processes, but is also very highly concentrated in volcanic outflows of various kind, both on the surface, on land surface, as well as in the ocean. So mercury comes from the magma into the earth, into the biosphere, and bacteria can methylate it, and they can also reduce it. And I'll come to that more at the end. But it does go around through all of these processes. Ozone interacts with both forms of mercury and can return it to soluble forms. And then there's industrial uses over here, which include local contamination from various processes to land and water, and also to the atmosphere. So that's it for the ecology part. Let's talk about cell biology. This one is to scale. These little molecules in this little diagram are actually to scale. This was done by David Goodsell, who's a crystallographer and artist at the Scripps Institute. And the various categories from the outside of the cell are proteins that occur in membranes, the ribosomal proteins that make other proteins. There are cytosolic enzymes. This is the cytosol. It's packed full. A cytosol is not just a glass of water with a few proteins floating around in it, okay? It's almost a jelly-like substance in our cells as well as bacterial cells. So here are the ribosomal and the cytosolic enzymes and chaperones, the proteins that help other proteins get folded. Here is what bacteria have: a nucleoid. They don't have a membrane around their nucleus, but it's an organized structure, and it's organized by proteins and also by RNA. And so the nucleoid also contains proteins. Nope. Oh, I see what it is. If you touch it more than once, then it moves. Let's try to learn that, Anne. Okay. All right. So the compounds that we've used primarily in our work are inorganic mercury, mercury chloride, mercury nitrate, and so forth. And phenylmercury acetate and ethylmercury thiosalicylate. And those are two organomercurials where the mercury is covalently associated with carbon. We don't use methylmercury in our work simply because we make use of analytical services that are outside of our laboratory, and they don't like it if you send them things in methylmercury. And so we do that in order to accommodate the analytical tools that we're using. But ethylmercury, as you may know, is what's in merthiolate, so that is important. And but it's an acceptable antiseptic, and so it's not something that we have to, it's not a no-no to send it out to a big service operation. Phenylmercury acetate was used as a disinfectant in paints and in feminine douches and in contact lens solutions for many years. Most of that's been phased out, but anybody who's gotten an injection lately knows, gotten a vaccination lately knows when you read down through what's in there, merthiolate is still in all adult vaccines. Though phenylmercury is not so much used in those anymore. So, okay, how does mercury kill stuff? This is the reductionist, right down at the nitty-gritty. We're talking about cells are made of living molecules, make up living cells, and so how does that happen? This is the equation of mercury with some counter ion. Mercury is positively charged with some counter ion associating into some stable complex, varying degrees of stability. And it's not doing this. Mercury nitrate is its most loosely associated form. It's almost like sodium and chloride in salt are very loosely associated, but the mercury compounds, the ones that are over at that end of the periodic table, they like to get very chummy with ligands, but not every ligand. And the point that I would have made more clearly with that is that as you move down and over in the periodic table to mercury, you get stronger and stronger associations. And so if you move over to I'll try this again. If you move over to the halides down here, chlorine and bromine and iodine make very strong associations, and these numbers are in logarithms, so it's 10 to the power of, okay, so very seriously higher associations here. And these are the organomercurials, methyl and phenyl. They tend to associate less actively with any counter ion, even chlorine, because they are also sharing electrons with the carbon atom. So, but the key point for biology is that we have monovalent thiols in biology, cysteine and glutathione. Cysteine's amino acid, and glutathione is a tripeptide. And we also have selenocysteine. Selenium is a different element in that same period with sulfur. Huge high affinity itself for mercury. So selenocysteine just substitutes a selenium for a sulfur than cysteine. And these association constants go up many orders of magnitude in terms of the association of mercury with monothiols or monoselenols in the cell. Selenium is rarely used in biology, but it's used in extremely important circumstances, which you can ask me about later. However, if you provide a bivalent chelator, which means this is the actual backbone of the molecule and it has two cysteines or two sulfur groups hanging off of it, that complex, which is called a chelate, will grab mercury very hard, very tightly. And you're familiar probably with compounds like DMPS and DMSA and Boyd-Haley's NBMI compound that are specifically designed in order to chelate, well, the latter is specifically designed in order to chelate mercury very tightly. And these are the highest association constants that you see with mercury. So you need to have, and the only natural bidentate compound is lipoic acid, and it's a very rare compound, and it has been used in natural detox of mercury, though the data on that are mixed. So this is the point that different monothiols have high affinity for mercury, and they can all kind of exchange rapidly. So if you start out with one monothiol, one monothiol associated with mercury and a second one comes in, it spins around through a tricoordinate complex, and it, just reassociates with whatever's there. Okay, so you can chase mercury off. So it's very kinetically labile, even though I just showed you that it's thermodynamically very stable. It's just not going to go and let chloride take its position. Sulfur isn't. But if you put another sulfur in there, it'll just spin around and the sulfurs can replace it. And that's one of the scary things about mercury in biology, because there's a lot of free thiol sulfurs floating around. However, in proteins, you often have adjacent cysteines. So cysteine is an amino acid that occurs in important places, usually in some kind of enzyme active site. So when you hit a cysteine, you're hitting something important. It's also used structurally to disulfide bonds make a kind of a nice link in proteins to establish their configuration overall. So when that happens, when you have adjacent cysteines that get mercury, the reaction is very much in that direction. It's not going to come off if you add a monothiol. So monothiols, that is all we got going for us in our biology, are not going to readily be able to release mercury from from a chelate, from bis coordination chelate in a protein. We looked at some processes that are disturbed in the bacterial cells. The bacteria have electrolytes just like we do. They have sodium and potassium, and they also use magnesium, and the electrolytes are sodium and potassium. And what we found was, looking overall at the effects of mercury exposure, and these are various mercurials, the black line is no exposure, and then phenylmercury and methyl and MT right here means merthiolate, and then even less mercury chloride and quite a lot of mercury chloride. And in terms of the amounts, there was no statistical difference in terms of the amounts or in the cell. The cell wasn't dumping any of these metals because it was exposed to mercury, all right? Except in the case of the two electrolytes, potassium and sodium. And there was a statistically significant effect of a difference in them, in that mercury exposure to any of the mercurials pretty much decreased the potassium content in the cell, and it increased the sodium content. And this is because bacteria have a proton balancing system to keep the protons inside the cell correct. And the pump that is the potassium proton antiporter is very mercury sensitive. It actually is listening to glutathione. That pump listens and it wants to have it glutathione, not disulfide. And mercury joining two glutathiones to get together structurally looks very much like a glutathione disulfide. And so that pump got slapped, and its regulation caused the cell to lose potassium. And sodium, the sodium pump listens to how many protons are in the cell. The protons were flowing in too fast, and so the sodium pump came up and started pumping sodium in. So that's a process that happens in bacteria, and said that even things that are not obviously related to metal transport or metametabolism, but to the glutathione status of the cell can be impacted. Now, the other thing that we were interested in these bacterial cells is what happens to proteins that have iron in them. Because proteins that have iron in them often hold that iron in two ways. One way is with heme, which is a great big complex carbon molecule that holds on to iron, and it holds it very ably and very stably. The other way is a more primitive way, and those are called iron-sulfur proteins. They're primitive, but they're very widely used in cells. We still use these for very important processes. And so we wanted to find out what happened when you added mercury to, let me continue to try to use this. Sorry about the guys on the other side. In the unexposed, this is the unexposed cells, and what we're watching here is the amount of free iron, and I won't go into how we do watch that. That's a well worked out biophysical process. If you add 4 millimolar hydrogen peroxide, the cell loses a little iron. It's got some pretty good protective measures against hydrogen peroxide. And butyl peroxide, butyl peroxide, 8 millimolar, also pushes a little bit more out. And then when you add merthiolate does almost as well as, at 160 micromolar, does almost as well as 4 millimolar hydrogen peroxide. And you go up with 40 micromolar, that's 100-fold less PMA than hydrogen peroxide, and you get a pretty big bump. And then with 16 micromolar or 80 micromolar mercury, you can go up, and this is much, much less than 4 millimolar hydrogen peroxide. You can go up and release close to 80% of the iron that's in the cell becomes free iron. And that amounts to pretty well knocking out the iron from all of the iron-sulfur proteins in the cell. So the cells are pretty sick at this point, but they're not. They're capable of recovering from this much material, but it's this much loss of stable iron. And the structure. This doesn't look like heme. This is an iron-sulfur cluster, and that's what that looks like inside the protein. It's held by cysteines, and it also has free sulfur molecules that are stuck in there. You have to make that with a whole bunch of other protein specialists that know how to make that and stick it into that protein. So it's an extremely complex process, but very widely found. And if mercury comes in, what happens is that mercury grabs onto a cysteine down there and replaces the iron ligand. And so it basically kicks the entire sulfur cluster, iron-sulfur cluster, right out of the protein. And that's what we're measuring in the graphic in the left. So that's two categories of difficulty that mercury can cause the cell at the molecular level with identifiable proteins whose function we can say, okay, we know mercury's hitting this guy. It's like we think of this as doing molecular forensics, where we're being called to a site where there was a mercury exposure and we see the victim lying on the ground. And our job as reductionists is to draw a little chalk line and then figure out where the bullet went in. Okay, so that this is part of why we do this, and the level of resolution that we bring to this is something that will be essential to know in terms of figuring out how the cell can recover from this process. So changing gears here is changing scale. It's now possible with modern tools of biochemistry to look not only at single proteins, individual proteins that have been altered by a toxicant, but to look at all of them at once. And this process is called proteomics. And this little cartoon indicates that a protein, of course, is a chain of amino acids. And there are enzymes that deliberately cut the amino acids at certain positions and break the backbone. You're probably most familiar with the enzyme trypsin. And trypsin is a protein. It is a protease. It's part of our digestive system, and it's actually quite neat in terms of where it cuts. It doesn't just chew the protein down into single amino acids. It goes in and it hits after every lysine residue. So it's got a special affinity for recognizing lysine residues, and it cuts those. And so you end up not with just a gamish of protein fragments, you end up with very nicely sliced up bits of protein that happen wherever there's an amino acid. If you happen to know the sequence of that protein, which we do for ever so many now, we know that this is in a database someplace. This is the sequence of the protein that ends up folding like that. And when you cut it with trypsin, all of these fragments, we know what to expect, okay? We know that we're going to expect peptides of specific sizes. So that's perfect for analysis by a technique called mass spectroscopy, because mass spectroscopy looks at things that are a different size. And you can shoot in your sample and it will give you a profile. And it says, okay, I see this many of this size and this size and this size and this size, and you know already from knowing the sequence in the first place that you expect peptides of certain sizes, and this is size. It's called M per Z, but you can think of it as a size. It's got so many amino acids in each fragment. And so when you actually look at your data, when you chop that up with trypsin, you'll have a player at each one of these same positions. Now you won't get 100% of every one of them because, as we say, not every peptide flies well in mass spectrometry. Okay, so some peptides are easier to see than others. But you'll get a distribution, all of which is always occupying particular mass here. Okay, so the mass is going to be based on, and that's what you go in and you look for. So you can match them, and you say, okay, I see this protein, and I see this many copies of it. You can actually see how much of the protein is being made, and to some degree quantify it with standards. Excuse me. So you can identify the peptides and therefore the proteins. Now, if you have got a protein that's been modified by mercury, covalently, mercury is going to be very much attached to that protein through a cysteine. So you know what to expect, that you're going to have a different size fragment for that particular peptide. Instead of having a peptide at, say, some position back here, this is an increasing size, you're going to have another peptide that has a mass of 200 Daltons. It has increased by the amount of mass that one mercury atom would provide. So that's 200 Daltons, okay? So that fragment will be that much bigger. So the mass spectrometer can see that. And when you see that, you know, that particular peptide is gone. I can no longer see it now. Instead, I'm seeing something that has a 200 Dalton increase. It is possible for it to have occurred, a 200 Dalton increase to have occurred for some reason. But one of the neat things about mercury, and in fact lots of the metals, is that they have multiple stable isotopes. So that unlike things that we're very familiar with, good old hydrogen only has a little bit of deuterium in it. And good old carbon only has a little bit of carbon-13 in it. It's carbon-12, and this is, and the same goes for nitrogen. And so those elements are pretty homogeneous with respect to their mass. But the metals, and many of the metals, and mercury in particular, have a wonderful spectrum of stable isotopes, not radioactive isotopes. These are always there, okay? And so there are, this, I think, there's 6 of them. There are actually 7. There's, yeah, 196 is a small one. But even though the average mass overall of all mercury in nature is 200.02, I think, Daltons, there's this distribution of metal atoms out there. All of them are chemically mercury, but they differ by 1 proton in their nucleus. Okay, I'm sorry, not one neutron in their nucleus. Okay, so they're very different in terms of their mass as far as the mass spectrometer is concerned. So these differences are capable of being seen by the mass spectrometer. And so if you find this peptide that has an increase of 200, if it only had an increase of 200 for some other reason, it would still have this kind of a distribution of its carbon, nitrogen, hydrogen, oxygen, and sulfur. But if it's mercury, it's going to not only have this mass distribution that is influenced by having all of these stable isotopes attached at any place, or in any given peptide, and it'll look like that. So you can make an unequivocal identification of a mercury adduct, as we say, to a given peptide, which means that you can go in and count what really happened after you expose the cell to mercury in terms of its proteins. And this is a profile of E. coli's proteins by functional class. So if you look down here, there are all the functional classes across the bottom here that proteins play a role in the cell. They include things like translation, which is the ribosome being able to make proteins and so forth. And I'm going to take you across this with another slide in a moment. But you can see that they're not all equally hit. The colors here means that this is the percent of a given group in the cell. So the blue thing tells you how many proteins E. coli even has in that category. And so that's blue means, 4% of E. coli's proteins are involved in translation. The red line tells you how many of these we saw in our experiment at all, modified or unmodified. Were we doing good proteomics? Did we see a lot of them? Did we see them proportionally? And so that green line tells you, okay, Summers et al., how many of these did you catch? All right. And then the red line tells you, of the ones you could catch, how many of them were modified by mercury? So that differs in various categories for several reasons. But this is the definitive tool to use for that kind of assessment. And so you can see that there are some things that are modified very extensively, that the proportion of them, the modification proportion, means practically everything in that category was modified. And so I'll take you through that. And so the bottom line was that we saw 307 mercury-modified proteins out of E. coli's 400-odd proteins. Incidentally, E. coli doesn't express all of its proteins all the time. It's a simple little bug, but it can turn things off. Okay, and so it's generally expressing about 2,000 proteins under the growth conditions that we used. So of those, we saw about 1,500, about 1,600 of them. And of those that we could see stably modified, we saw 300. So who are those? Come on, do that for me. Okay, so you know about ribosomes that make proteins. Transfer RNA is very important in that process and has to be made. And these are these other categories of proteins are elongation factors and initiation factors. So they're key to being able to make a protein. You also have to be able to make RNA to encode the protein. So that this is transcription. This is RNA polymerase, which has 4 subunits in bacteria, and all of those were hit. The sigma factor and the R termination factor, those are other factors that have homologs in humans but are not identical, but they are important in this transcription process. ATP synthase, okay, this is the energy-making entity of any aerobic organism. In us, it's in our mitochondria, which are little ancient bacteria that invaded our ancestral cells and made it possible for us to be big air-using human beings. Okay, we have this enzyme. ATPase was one of the most highly hit of our systems. 3 of its subunits, and then the others are involved in a pathway that you probably will break out in a sweat if I mention that you probably had to memorize the Krebs cycle or the TCA cycle when you were in biochemistry. That's a dreaded cycle that processes both energy, is connected to both energy as well as to intermediary metabolism, to making the small components that go into making proteins and so forth. And it was also very heavily hit. Amino acid biosynthetic pathways themselves are heavily hit. And so that's a great many different enzymes involved in synthesizing each of those amino acids. So those kinds of changes would naturally, proteins in those categories, proteins getting hit in those categories are naturally going to disrupt the cell processes. And we'll come to that in a minute when we talk about transcription. So this is just a listing of the proteins that where mercury has displaced the iron cofactors. So I told you about the iron cofactors and how they're important. And now this is kind of a sort of a taxonomic presentation of those. We know where the cysteine hit. Okay, we know what kind of an iron site it was, and we know what the adduct was. Did it get hit by mercury mostly, or was it more messed up in the phenylmercury experiment? And that actually does differentiate some of these proteins and some of these sites. Some of them are more sensitive to organomercurials, and some of them are more sensitive to mercury. The functions that we're talking about here, the functions are energy production, okay, that's the involvement of these proteins. Amino acid metabolism, as I just showed you, these are some of the guys that got knocked out in that category. Inorganic ion metabolism, so this is getting sulfur uptake into the cell in this case. Lipid metabolism, you have to have lipids in order to have a membrane. And so lipids, in fact, are extremely important in the structure of any cell. And this is a regulatory protein. Regulatory proteins are not very abundant, but we did see quite a lot of them that we could hit with mercury. And then proteins get modified, even in bacteria, proteins get decorated sometimes with sugars or with lipids and other things. And so those are processes like that we have in our own cells. And what's important about this particular subset is of the proteins that we saw, how many of these things do we have homologs in our own cells? And how close are those homologs? Is the position where the bacterium was modified similar to the position in our very own proteins? Okay, because our proteins, particularly, as I'll point out in a moment, see the M up here in the corner? I think that turns red in the next one. Yeah, it's the same slide. That M up there means those proteins actually are in our mitochondria. Okay, so these are picked off of our list of hits in the bacteria. And we look to see, this is the degree of peptide homology, just how similar is that peptide, the whole peptide that the cysteine is in. So the cysteine that we saw modified is a cysteine that occurs in a peptide in these proteins in us, okay? And those proteins, have the similarity of that whole region of that protein ranges from 55% to, in one case, 94%. So these are, in principle, targets that could be identified, could be seen in our own cells of mercury exposure. And one of the reasons that we're driving in this direction is that we want to be able to find biomarkers of mercury exposure. And that's hard to do because any kind of exposure biomarkers that you can find for almost anything, that's the key to establishing toxicological profiles for danger and so forth. And it's very fuzzy, as you may know, for mercury. So if we can go in and say, okay, we've taken blood from this particular human or taken a tissue sample from this particular human, and we can pick out the transaldehyde and we can take that out with antibody. We can just grab that protein out. We don't have to do mass spectrometry on everything. We can just grab that protein with an antibody to transaldehyde and then do mass spectrometry on that. And we're looking at that protein to see whether or not it has mercury on it. And so this will, to some degree, be a beginning of figuring out how different humans get poisoned in different ways and what their genetic vulnerability is. Nailing it right down to, yes, you have mercury on this protein in your body, which is a whole different thing than just saying, how many fillings do you have? Not to say that having one protein or another is not it is going to make mercury in your mouth okay. It's not. But I'm sure, as many of you know, the whole business of getting mercury disapproved to a great degree depends upon being able to do the biology underlying it, where we can show, yes, this is the reason. Just as Dr. Cantor was talking about earlier, the genetic connections to mercury intoxication, like the genetic connections to the periodontal processes, are going to be very important going forward, not only for regulations, but for thinking about how we can design protocols to help this human or that human. Personalized detoxification has got to be a part of personalized medicine. So, how are we doing? Okay. So we wanted to ask, knowing that these things had happened to the proteins in the cell, how is the cell responding? And as I alluded to, and I think Dr. Cantor also mentioned the subject of our transcriptomics, is what the general area is called, of finding out what a cell is doing when you hit it with mercury. So in these experiments, we had cultures of E. coli growing in a little glass flask, and when it grows and you don't mess with it at all, it'll double about every hour like this. But if you hit it with mercury at this point, it's been growing, so this is the beginning, it's kind of the middle of the growth period. Hit it at the middle of the growth period with mercury. In the red line is mercury chloride, and in the green line is phenylmercury. And you can see that they are recognized, just in terms of the behavior of the cell, in terms of its doubling and growing, they look different, okay? And we did these experiments on a short term because we didn't want to get into a lot of cell division issues. And so we stayed at the level of between 1 and 1.5 doubling times for the cells. What we were looking for was how long does it take these guys to recover this growth rate. So you have a growth rate, and that's your figure of merit over here, and you're waiting for, well, how long does it take these guys to get back into that growth rate? And you can see clearly that the PMA-exposed guys return much more rapidly than the mercury-exposed guys. And that was something that we saw consistently throughout this. And at these time points, we took samples, physical samples of the cells, and isolated messenger RNA. Now, there are lots of RNA molecules in the cell, but you may remember that messenger RNA is the RNA that goes and encodes proteins. So it says, this is what I want to do now, okay? I want to switch. I need to do something different. If you see a change in messenger RNA. So it's possible to remove the other RNAs in the cell and look only at the message. And that's then sequenced by base sequencing, and we can tell what it is. Now, has anybody in the room ever seen a heat map before? Anybody? A heat map? Yeah, okay. So this is a heat map. It's a way of showing an enormous amount of data and boiling it down to just what's really going on. What is the most important function that is going on? So let's go through this like this. So over here, this is the heat, okay? If it's yellow, that means there's a whole lot of stuff going on, that there's lots of intensity in that particular band, okay? And as you get down into the middle and where it's dark again, that means there's not a lot of difference here. And down here says, these guys are getting turned off. So let's go back to this. What you're doing in this comparison is you have messenger RNA isolated from each of these conditions in each of these different time points. And the figure of merit, the tool that we're looking at, is a given messenger RNA compared in this condition here to the guy that didn't get hit. So that's our baseline. We're comparing, everybody's growing, but we're comparing the guy that's not hit at all. He's just fine. We didn't do anything to him. How different is the messenger RNA in this guy as at each time point compared to this? And the same thing for the red line, okay? And that's what a heat map generally is showing you. It's talking about a difference between one condition and another condition. So this is the heat. That says it's really going on, and this says, hey, it's turned off compared to that other guy that didn't get hit. And these are the conditions. This is mercury down here at the bottom, and also PMA down here at the bottom, okay? And these are log-fold 2 differences, but don't worry about that. The numbers are bigger or smaller, all you need to know right now. What are these things over here? Those are all the genes in the cell, okay? So each line going across is a different gene. And you say, well, I can't see that. And that's true, you can't, because that's a very large number of things that were changing, like 1,500. So the point here is a qualitative point, and this is an important one to make from heat maps generally, that the changes at the time points for mercury don't look like the ones for PMA. Some of them overlap, but PMA has a whole swath of genes up here that are turned on and then turned off, okay? And over here, mercury has some of those genes turned on, and then not even turning those genes on. And still out here at 60 minutes, I'm still turning on some of those genes, okay? So that the profile overall of PMA exposure suggests that it's just like the line. I'm getting over this exposure really fast, okay? And so what this shows is that while there is some overlap, the cell is recognizing mercury as a different biochemical toxin than organomercury. And that's important to know because there's a huge lore out there in human biology about, well, what's the most toxic form of mercury? Okay, and one of the reasons we did this was to help resolve that. So, gotcha. These are some specific functions that we looked at that have to do with, now this is teasing out specific sets of genes, and we just made a heat map that show us everything that's downregulated grouped together, show us everything that's upregulated, and you can see there are different categories that are downregulated, because these are the categories of things that were in the blue over there, and this is for mercury and for PMA, and over here you can see in the right-hand side, it's different categories. And among the important things to notice over here, this is the electron transport complex and ATPase. The cell is shutting it down, okay, when you hit it with these mercurials. And it's doing it, in fact, with both PMA and mercury, turning the electron transport process off, okay. That's really bad. If you're going to recover, how can you turn off your energy generating processes? Well, the heart of that is the electron transport pathway, when it's not functioning correctly, is the source of reactive oxygen species. Because it's all the time interacting with oxygen. But if you don't do it just right, you're going to get superoxide. And you're going to get superoxide dismutase turning into hydrogen peroxide and then a hydroxyl radical. So it's very characteristic of a damaged cell in any way that it turns down the electron transport pathway. So that is one of the things that we were looking for as a hallmark here, because it's characteristic of mercury in mitochondria that one low-dose mercury hitting the eukaryotic cell, one of the hallmark phenomena is a burst of reactive oxygen species. So this is E. coli is doing the same thing, kind of thing that mitochondria do in us. And I'm going to scoot along because there's a chunk of this that I do want to talk to you about. And this is just kind of summary. It does matter from cell biology, proteomics, it makes a difference what you're being poisoned by, and transcriptomics. Now, this is the groups that have helped on this and the funding agencies that have contributed to it. But let's go to the last section and remind ourselves that in that big slide of the world that I showed you, there were fish, but there weren't any people, okay? And so it really didn't talk about the kind of exposure that people have. But we know people have mercury in their mouths, and that's what we're concerned about. And so these numbers are a little bit old, but this is the exposure to mercury that's available from dental amalgams. And there's thousands of people out there walking around with grams of mercury in their heads, as you well know. And if we stopped doing all mercury fillings, not we, but you, stopped doing all mercury fillings today, they'd still be out there, okay? And so thinking about doing something about those folks is still important. Yeah, these are numbers that I think you're probably familiar with. And there's been associations with many different kinds of chronic diseases. You surely know the work of Dr. Dan Lax, UCLA, I think, on the association, and also Mike Pollard with autoimmune disorders. And what we're concerned about ourselves is an ecosystem that's inside the body. It's a much more popular now. 10 years ago, if I had given the same talk, everybody would have blanked over just as much as with heat maps if I'd said something about the microbiome. The what? Okay, but you can't hardly open a magazine these days or scan your news in your phone without seeing something about a microbiome. And of course, we microbiologists plotted all that originally, and we're really glad that y'all have caught on. But it is the case that it is a tremendously diverse system. And the body's immune system also, 80% of the body's immune cells are in the GI tract. 80% of the serotonin receptors in the body are in the GI tract. There's a whole chunk of neural material lying just outside the GI tract that's referred to as the brain and the gut. Okay, so that all of this is next to something that amounts to between 2 and 5 pounds of a microbial community that's in the GI tract. Okay, so we have to pay attention to that. It's like having a whole different organ that we can actually change what it does all the time depending upon what we eat and what we throw down there. So those commensal bacteria constitute a reservoir of antibiotic resistance genes. Antibiotic resistance doesn't just pop up spontaneously. I won't talk about the mechanics of this now because I'm running short of time, but it's carried by mobile genetic elements. Antibiotic resistance is on things that every bacterial cell has a chromosome, and if you have a point mutation on one of those chromosomes to antibiotic resistance, yeah, millions of generations later, that'll be a significant population in your body. However, if you have mobile genetic elements, which all of them do, those mobile genetic elements don't have to wait for daughter cells and daughter cells and daughter cells and daughter cells to become everybody there. They just share the genes, okay? And physically moving DNA from one cell to another in the form of these small plasmids. And you can see them on the gel. They really are true. How many of you have heard about the escape pathogens? Is any on anybody's? Okay, escape stands for is the first letters of the names of all of these bacteria. These are the major targets of multi-antibiotic resistance concern, and every one of them, in every single case, is the antibiotic resistances in these bacteria are carried by mobile genetic elements. Not single mutations on the chromosome, though those can occur. They are very quickly moved up to be on mobile genetic elements that can then carry them anywhere at all. So that's a big problem. And the mercury resistance locus that we study is on a mobile genetic element. It always has been. And it sits right next to a major engine of multi-antibiotic resistance generation that I won't go into right now. It's called an integron. It can have 9 or 10 distinct antibiotic resistance loci in it. And it sits right next to the mercury resistance locus. So that's it. We'll talk about that. We found many years ago that human beings had mercury resistance in a distribution. And so we looked at various human being samples and how much mercury resistance was in a human microbe sample, going from none to 100% of them were mercury resistant. And what we found was if you had lots of mercury resistant bacteria, you were more likely to have multiple antibiotic resistance in those bacteria. And correspondingly, if you had very little very little mercury resistance in your bacteria, you were more likely to have no multiple antibiotic resistant bacteria. And this is 1993. Okay, and we did work on monkeys that was published where we had fillings put in. This was with Fluoride-Free Murray Vimy. And this is the mercury coming out and goes up pre-fillings. It goes up 5 orders of magnitude in the GI tract over a week. It's millimolar levels of mercury in the GI tract over a week. And then it gradually comes down again. And in 2 of these monkeys, the fillings are replaced. There was a spike for that reason, and then it went way, way down. And for these monkeys that didn't have it, and then it goes back all the way down to background. It always stays. It's actually a selective level. And the key thing here is that we found was that the mercury resistance in these animal bacteria, that we looked at many of the bacteria, there was some detectable there beforehand, but when the mercury went in week 0, which is down here, the mercury resistance came up. Increasingly, the mercury-resistant bacteria were as many as 60% of the bacteria in that category that we were looking at, which is not 60% of the gut, but 60% of that category of bacteria, which is an escape bacteria, were resistant to mercury, and they were also resistant to the couple of antibiotics that we had the money to survey at the time. And subsequent work showed, yes, these are carrying an integron. The latest thing on this is a paper in 2015, not done by us, but done by some guys in Sweden. And this is a big survey of all the genetic information that's out there in the big databases. And they asked, how often do you find a metal resistance along with an antibiotic resistance? And this is what they found. You got some copper resistance out there, and the orange stuff, and this is copper as well. And you don't see it associated with these little blue things. The little blue things are the antibiotic resistances. But the little blue things on this side are completely surrounding this particular metal, and this metal is mercury. And so these are the genes of the mercury resistance locus. And they're completely surrounded by these little individual antibiotic resistance genes. And so this is what's really out there as far as the other omics, genomics, and informatics. This is the data from GeneBank that says, yep, It's out there. These guys basically identified the locus that we work on, TN21, informatically. They just asked the database, and the database is rich enough right now that you can see a signal like that. So I have been, I'm not going to go to silver. I answer questions later on about silver, but we will wrap it up right now. And with thanks. And what's in the works, some epigenetic works, and also looking at transcriptomics for cells that have got the mercury resistance operon. So thank you very much.